From computer-guided surgery to medical imaging diagnostics, Ron Kimmel has a sharp vision for the future of healthcare. Kimmel is a professor in the Henry and Marilyn Taub Faculty of Computer Science, where he holds the Technion Montreal Chair in Sciences. His work covers a wide range of areas, including shape reconstruction and analysis in computer vision, image processing, deep learning of big geometric data, and computer graphics. Ron's research is focused on adopting tools from modern geometry to overcome some of the most fundamental yet challenging obstacles when it comes to the reconstruction, analysis, and synthesis of deformable shapes. His goal is to develop computational tools that lead to real-life applications in the healthcare industry and beyond. Tamar Koren is an MD Ph.D. candidate at the Rappaport Faculty of Medicine at the Technion. After graduating cum laude from the Bookman Mehta School of Music in Tel Aviv as a violin player, she began her medical studies in 2013 at the Technion. In 2015, she joined Professor Asya Roll's lab as a research assistant, where she worked on her Ph.D. dissertation on encoding immune information in the brain. Her research revealed a psychological basis for the psychosomatic phenomena through the discovery of immune memories in the brain. The groundbreaking findings were recently published in the scientific journal Cell, sparking attention from both the scientific and medical communities. Tamar's research could lead to better clinical outcomes and new treatment options for certain diseases in the future. Associate Professor Shai Shen-Orr is on a mission to decode the inner workings of our immune system. The computational biologist currently leads a team of researchers at the Systems Immunology and Precision Medicine Laboratory in the Technion's Rappaport Faculty of Medicine. Shai specializes in applying computer science and information technology to the fields of biology and medicine. It's all part of an effort to deepen our understanding of the immune system and its relationship to disease and aging, ultimately leading to advancements in genomic medicine. Shai is also researching diagnostic biomarkers that more accurately evaluate human health. His work could lead to major advancements in the diagnosis and treatment of immune-related diseases.
You're going to now see how bad I am at following directions. I'll also start out by telling you I wish I understood biology or anything in the magic of what these people are doing. I don't. So I'm just going to first ask a real expert from the Technion to come up, and I'm told you're going to give us a talk. No, Tamar is going to give us a talk, and then I'm supposed to act like I can ask an intelligent question. I'll try. But I'm hoping that you will convey what you're doing in a way that they and I will understand. Tamar.
So first of all, thank you. It's a great honor for me to take part in such a wonderful meeting and in this honorable panel. I'm in the MD PhD program in Asya Roll's lab, and I'll be mostly talking about my work I do in the lab, dealing with basically trying to harness the brain's potential to improve health, and I'll try to deliver this idea to you here now. So I'll start with something very general, so bear with me, I do have a point. So imagine that a friend with whom you just went out last night texted you that he or she has COVID. How long will it take you to feel the itch in your throat, a slight headache, and maybe even your temperature rising? Now, for many of us, including myself, just the anticipation of a disease can manifest in an actual illness. Now, we often disregard these feelings and blame them on our powerful imagination, but maybe if a thought can actually, maybe if the mind can persuade us that we feel something, is it really just our mind that is persuading us, or something is really happening in our body? Now, this phenomenon of mental states that are expressed in bodily manifestations is not new and is widely acknowledged in the medical world, often regarded as psychosomatic or functional disorders. One early and interesting example of this was shown by an American physician named Dr. John Mackenzie in the end of the 19th century. And Dr. Mackenzie had a patient that was allergic to the pollen of flower. And he saw that when he presented this patient with an artificial flower, she immediately reacted with sneezing, coughing, itchy throat, and basically all the mucus accumulating in her nose, basically all the symptoms that resembled an actual allergic response. So what was it? If a thought triggered by an external cue, like an image of a flower, can manifest in signs of disease, can really the mind cause or generate? Does the mind have the power to generate an actual illness? Now, another example that was made through experiments in animal models several years later showed that just like the classical Pavlovian learning, the body can also learn certain immune responses, what was called immune conditioning. Now, according to this immune conditioning paradigm, the body is, after you give several administrations of an immunosuppressive drug, together with a sweet tastant like saccharin, it is sufficient to administer the saccharin alone in order to induce immune suppression, just as if we administered the drug itself, meaning that the sweet tastant, the saccharin, has functioned just like Pavlov's bell. Now, these examples all kind of proves us that there might be some kind of representation of immune responses in the brain that may be even recalled by some kind of an external cue, like an image of a flower or a sweet glass of juice, for example, or a sweet tastant. And this, if you think about it, it kind of reminds us of how memories work, like a certain sound or a scent can make us recall a childhood memory, for example. And this is exactly what we wanted to figure out in Assero's lab. Are there immunological memories in the brain? Now, how do we approach such a question? So we decided to use a tool that was widely used in memory research studies. And this tool helps us to capture neuronal activity, specific neurons that are active during a specific time point, like an inflammatory response. But not only to tag them, but we could also express on them certain receptors that help us to manipulate them and control their activity, to see whether we can control their activity, manipulate them, and see if we can actually induce some kind of immunological effects. Now, we chose as our immune challenge that we wanted the brain so-called to remember is the model of inflammatory bowel diseases. And we chose this model in animals for several reasons, and not only because the gut is a highly innervated and immunologically active site, but also because we know that in several diseases that are affecting the gut, like ulcerative colitis or Crohn's, people that are suffering from these diseases often suffer from their outbreaks or their disease during times of stress or some kind of highly salient or emotionally salient events. So maybe these events or stressful times are playing as a trigger or the external cue for the recalling of their previously encountered inflammation. Now, the next question that we wanted to ask you is, where in the brain should we look? What brain region can be capable of encoding specific immune information? And we had we thought about a very promising candidate for that, and this was the insular cortex, or for short, insular. Now, the insular cortex is considered to be the site in the brain that is responsible for interoception. And interoception means that it integrates information regarding our physiological state, the inner state of our tissues and our organs. Or in other words, it is the brain region that is dedicated to know how do I feel. So if the immune state is such a pivotal, plays such a pivotal role in our physiological state and in our well-being, it is only reasonable that the immune information is also part of the information that the insula can integrate. So in order to understand that, we again used the tool that we decided to use in order to tag neurons that are active during colon inflammation, specifically in the insular cortex. And after the mice healed, we waited for them to heal and basically return to their healthy state. We then reactivated, I'll go back. We then reactivated the same neurons and looked at the immune response to see whether we could recapitulate an inflammatory response in the colon. And what we saw, as you can see here, there are two slides of colon tissues. From your right, you can see yes, it's your right. You can see the slice of a tissue, of a colon tissue from the group of mice that went through the insular activation. And from your left, you can see the slice from their non-activated controls. And the little red dots that you see there are immune cells. And when we measured these immune cells, we saw that there was a twofold increase in the presence of these immune cells in the tissue after reactivation of the insula in these mice. And we saw it in all other inflammatory related parameters and subpopulation of active immune cells. Meaning that by reactivation, specific neurons in the insula in the brain alone can recapitulate some kind of an inflammatory state in the colon. So we next asked, okay, but how specific can it be? Is it just some kind of recapitulation of an inflammatory state, a general inflammatory state in the body? What would happen if we chose another inflammatory model? Could it also remember and recapitulate it? So in our next experiment, we chose a different inflammatory model that is affecting the abdominal wall, the peritoneum, but not affecting the intestine. And we did the same paradigm. We let the mice heal after we tagged the neurons in the insular cortex during peritonitis or peritoneal inflammation, and then reactivated these neurons. And what we saw that again, the inflammation reemerged, but this time it was specifically in the peritoneum, and it was specific to the type of inflammation that we induced. And also you can see here the above graph, the graph that you see over there, the first one, or the biggest one, is what basically represents the inflammatory response in the peritoneum. And when we looked at the colon, there were no effects. None of these reactivation of the insular cortex did not induce any effects in the colon, meaning that the recapitulation could be actually specific to the peritoneum if it was previously captured or tagged during peritonitis. So it was very exciting to us to see that we could actually recapitulate different inflammatory responses in different sites in the body. And we wanted to understand, how is it possible? How can we induce from the same region in the brain different immune reactions at different sites in the body? And there are several pathways in which the brain can regulate and induce different effects in the periphery, whether it's through the neuronal pathway that comprises of the parasympathetic nervous system and the sympathetic nervous system, or through hormonal pathways, through hormones. So we decided to look at the potential neuronal pathway, that maybe through this pathway the insular cortex speaks to the inflammatory sites, the peripheral sites. And for this we used the rabies virus, and this is the same rabies virus that you know that can infect a peripheral site in the body and go all the way through neurons all the way to the brain. So we utilized this capacity of the virus in order to try to track the neuronal pathway by which these potential neurons can actually communicate from the brain to the peripheral sites. So we injected the rabies virus to the colon wall of these mice. And as you can see here, this is in green. You can see the neurons that were infected with the rabies virus in the colon. And when we looked at the brain, specifically in the insula, as you can see here from the yellowish stained neurons, these are the neurons that actually were both active during the colon inflammation and also stained with the rabies virus, meaning that the rabies virus actually reached the insular cortex to the same neurons that were active during inflammation, meaning that there is indeed an anatomical connection between these neurons to the inflammatory sites that can be actually mediating such immunological effects after neuronal activity. And we saw that this neuronal pathway also go through two of the regulation centers of the autonomic nervous system, both the parasympathetic and the sympathetic system. And probably this suggests there's some kind of balance between these systems in order to mediate the immunological effect of the insula. Okay, so I'll just recap. So we saw that we could reactivate only by neuronal activity in the brain specific inflammatory responses. So our next question was, if we could reactivate inflammation, could we actually alleviate inflammation if we inhibit insular activity? So in our next experiment, in the last experiment, we inhibited the insular activity during colon inflammation, and we wanted to see what happens now in the colon. So we looked at several inflammatory-related parameters, both the length of the colon, the spleen weight, and the number of immune cells in the tissue. And we saw that in all of these parameters showed a decreased sign of inflammation after we inhibit, and specifically in the group that went through insular inhibition. Meaning that indeed, by inhibiting neuronal activity in the insula, we could not only alleviate and decrease inflammation, we could also see less inflammation and clinically wise. And these results really excited us because it kind of meant that there might be some clinical relevance to these manipulations, specifically in patients that are suffering from autoinflammatory diseases, chronic autoinflammatory diseases like not only ulcerative colitis and Crohn's, but also rheumatoid arthritis, psoriasis, lupus, and even multiple sclerosis, all of which are diseases, incurable diseases that the patients that have them are suffering from debilitating outbreaks of their diseases, mainly following emotional states and stressful events. So we thought maybe if we can inhibit aberrant activity in the insular cortex of these patients, which we know some of them do have increased activity in the insula in these patients, maybe we could alleviate their symptoms and even maybe prevent their outbreaks of the diseases. And we know today of several tools, non-invasive tools that can allow us to give this way of inhibition to these patients in order to inhibit or manipulate their neuronal activity, and maybe in the hope for a better use of the brain immune connection therapeutic or curative potential. So I'll finish with that, but I first want to acknowledge all the people that without them this work couldn't have been made. First of all, Asir Rolls, my advisor and the one that gave me the opportunity to work on this project when it was only theoretical. My incredible co-lab members that you can see here. Of course, the funding of this work. And I would also like to mention all the families and supporters that maybe some of them are here in the crowd that support the MD Ph.D. program that can allow us, students like myself, to fully dedicate their time doing basic and clinical research during our medical studies. So thank you, and enjoy your rest of the day.
The pressure is on. I'll start by saying I don't have a very technical question, but the very first thing you said, which I was unaware of, and I'm assuming most people are unaware of, back in 1896, a doctor, somebody who's here to help us, figured out how to give somebody the bad results of sneezing and allergies without even exposing them to pollen. Counterintuitive. Mm-hmm. However, it's well known to anybody that's looking at medical research, they'll always do double-blind testing. They always have to have a reference group. And what's called the placebo effect has been known in every study. Hey, if you tell people they're going to get this drug or this treatment and it's going to help them, but you have to take half of them and tell them they're going to get it and all they got was a placebo, sugar. Almost without fail, the placebo group does much better than a person that's not in there at all. They probably aren't going to do as well if the drug really works or the therapy really works. But you've sort of given credibility to the fact, and maybe we should stop calling it the placebo effect. And if you optimize that piece of the constituency, is there anything in all of your research that makes you go back and look at this and say, it's not a placebo effect at all, and maybe you could learn from what has gone on so consistently in that area that you can optimize therapies that don't require anything except what was formerly called, dismissively, the placebo effect?
Yeah. Okay, I'll try to answer this question. Well, the placebo effect is something that it was also investigated in our lab, in Astra's lab, and they saw that the placebo effect is regulated through a different kind of brain region, which is the ventral tegmental area, the VTA, which is it's part of the limbic system or the reward system in the brain that is basically responsible for a motivational behavior. And what they showed in the research that they've done, that by reactivating this reward system, you can actually show not only the motivational behavior, but also to boost immunity through these neuronal pathways. And this is also a project that Shai was involved in. And what we were talking about, and I think it does relate to the placebo, but it, I think, gives another perspective because it's something that was very specific. And it showed that there are specific neuronal pathways in the brain that can encode memory of immune responses. So the placebo effect can work even when you receive the first time your drug or the placebo drug. But what we're talking about is once you were exposed to, say, like a pathogen, for example, and later on heal, and then maybe the neurons that were active during this inflammatory or infection, when they reactivate for some reason, for some kind of external cue or internal cue, then they reactivate the previously encountered response. So I think these are the maybe the differences between the placebo effect, which can be made in any human, even without previous exposure. But here you have the memory component, the mnemonic component, that is crucial in order to reactivate specific inflammatory responses. So I think this is the...
I'm going to propose that there's some sort of dominant insular pathway in these so-called placebo patients. I'm a clinician.
Oh,
I see it
really?
all the time.
Wow, interesting.
So that's what once you map out that insular region of the brain, you get a pathway going to the bladder or the bowel or the skin. That's going to be the dominant pathway, and that would probably be incorporated into our medical education. But right now, chronic fatigue syndrome, we don't know what it is.
So you say it synthesizes these neuronal pathways in order to achieve different inflammations and different...
Yes, yes. Mm-hmm. You outlined it beautifully.
Wow. Interesting.
Thank you.
Thank you. Thank you very much.
Ron, it's up to you.
Okay, thank you. So, first of all, I'm honored and really happy to be here in this beautiful place. And I will try to talk about computational oncology or computational pathology, or more exactly, how can we use computers in order to help us analyze pathology. I will talk exactly what it means. So how to use modern computational methods in order to help traditional medicine. So first of all, the engine behind our technology, and this is probably the most important part. These are my students, some students. There are several engineers that we are using and physicians. I will just mention Gil Shammai. He's a postdoc in my lab, and he's basically running this thread of computational pathology. Next are our main clinical collaborators, and I would mention probably there are many head of departments of oncology and pathology in Israel. Edmond Sabo from Koren Hospital has been really, really useful helping us with high quality data, acquiring high quality data and defining questions. Noam Asna from Ziv Medical Center. Iris from Shiba, and many others. Okay, so what do I mean by computational oncology and pathology? So what would be our mission? Our mission is to leverage the data that is being available to us through samples of tumors, and then help the oncology to get more clever information about what to do with this information. So we would like to use computers in order to look at tissue slices and thereby help the pathologist define a better way of treating. So should it be chemotherapy or just cutting the tumor away or radiation? Currently we claim that we can do things, but we can improve them. So what is the current process? Assume that you are diagnosed with you feel that you have a lump in your breast. The common thing that happens is that the physician is taking a biopsy, say a needle sample. Then this sample is being delivered to the pathology department. What they do is they slice it really, really thin, like Swiss cheese. They color it with something which is called hematoxylin eosin. It's really cheap, it's really old. It's the classical way of being able to look at these thin structures, these really cell-level slice of the tissue. The pathologist then uses all his knowledge or her knowledge in order to decipher what he's looking at. So the type of cancer, how severe it is, which is called the grade of the cancer. And then this is the beginning of the investigation. Now, in order to really isolate the exact type of cancer, he or she needs to define, for example, the hormonal receptors that characterize the specific kind of cancer. To that end, what they do is they color it with a different type of coloring, which is called immunohistochemistry, or they apply something which is called next generation sequencing, look at the sequence of genes, and at the end of the day come to a conclusion. They provide their analysis to the oncologist, and then the oncologist look at all the data that he or she knows, browsing through the internet, consulting with friends, and eventually deciding about the treatment. Where do we come into play? My profession is computer vision. What is computer vision? Let me just give you a brief about computer vision. If you take an image and you try to improve it, this is called image processing. If you try to understand the geometry that is going on in an image, this is called computer vision. This is called the analysis. And if you do the inverse problem, taking geometry and trying to reconstruct nice-looking images, okay, you can process the geometry, and if you do the other way, this is called computer graphics. And this is my arena. This is where my students and I are actually spending most of our time. Now, recently, there have been some newcomers to the game. One of them is taking geometric structures and trying to push them into the real world. If you heard about Pokemon Go, this is called augmented reality. It's also useful in when you try to operate on somebody and you have the CT or MRI data and you are trying to project it virtually on the body and then know exactly where to slice, what to do the slice. This is called augmented reality. Or you can do the opposite. You can take things from the real world. Taking a geometry and photometry image like the lighthouse here and putting it in the virtual world, and this is called virtual reality. So I hope I did a little bit of order in what you heard about augmented, virtual, computer vision. This is my arena. This is where I spend most of my time in. When I came to the Technion a couple of decades ago, they told me that the holy grail of computer vision is to segment an image, to give a semantic segmentation of an image. In this case, for example, if I would be able to take apart this image into the fence, the lighthouse, and the buildings at the back, if I know how to do that, then I would have solved the most important problem in computer vision. Now, 10 years ago, there was a revolution in computer vision and, in fact, in all of computer science due to three scientists, Jan LeCun, Jeff Hinton, and Joshua Bengio. who basically revived an old technology which is called deep learning. Okay, so deep learning, and they have, I mean, in science we really care about how many citations we have. These guys have about million citations, or more than million citations put together. And I'm not sure which Joshua has more citation, the original or Benjio. Nevertheless, this revolution allowed us to solve really interesting problems, and one of them is related to what I'm going to talk about. So when we look at what is going on at the way that people are treated here, after doing some analysis of the biopsies that are taken from the skin, it appears that about 10% of the patients had to change treatment due to rediscovery of, for example, the estrogen receptor or the progesterone receptor that were wrongly diagnosed at the first examination that they went through. And the question is, can we help that? I mean, can we assist that? Let's focus on the H&E, on the very basic, simple and cheap first slice of the tissue that we are looking at. Physician can tell you about whether there is cancer or not, the type of cancer, and what is the grade, but they cannot really tell you what is the genetic mutation that caused this cancer, what is the molecular receptors that classify this cancer, what is the prognosis, I mean, what is the risk, and eventually what would be the best treatment. And the question is, can we train a machine, a computer, to look at these images and give you these answers? And let me just jump to the end and tell you that the answer is yes. I mean, we think, and we are actually verifying it statistically, but the answer at the end of the day is yes. So again, assume that you would like to verify that there are estrogen receptors. You recolor it with immunohistochemistry, and this is how it looks like. This is ER positive, and this is ER negative, and this is something that a pathologist can look at and determine whether this works or not. Can we look at the previous image and say if there is if it's ER positive or negative? So we train the system. What you see here is the same slice colored with the H&E, has the basic coloring, and the green spots, and there are also red spots, are the spots where our machine told us that these are the receptive fields, the numerical support that it needs in order to make a clever decision. Okay, so this is, in fact, we showed that we can find most of the biomarkers that are needed for further investigation as we look at this kind of H&E images. The more samples that we introduce the system, the better it gets. Until now, we were not able to saturate it, so the more data that we have, the better our predictions are. It's very important to, I talk to many intelligent people here in the crowd, and at the end of the day, you need to manufacture. You need to compare to yourself to the rest of the world. So compared to the rest of the world, we had really high quality and high quantity data, and this is done by a lot of legwork. At the end of the day, using the system, and this is a system, it's a black box at the end of the day. Also to me, we can decide, we can go for better treatment choices by just looking at the original H&E hematoxylineosine colored images. So we can do disease prognosis, the subtype within the cancer, the risk group, response to therapy. These are things that we can predict much better. In fact, much better than you would have given me the immunohistochemistry coloring as well. If you put the immunohistochemistry coloring as well, we can actually go further. So again, I can tell you a lot about our solution, but it would be too technical for me to dive into that. But again, believe me that it works, and I hate saying that, but it does. And at the end of the day, we have been the first to look at these H&E images and find the estrogen, the progesterone, HER2, etc. receptors in these fields. And in fact, I don't know how many of you heard about Oncotype. So Oncotype is this kind of way of looking of providing all the data that you have in breast cancer. So you give this company your response to HER2, which is this kind of biomarker, as well as 21 genes. 16 refer to classical pathologies like BRCA, etc., and 6 for reference. And they put everything in a computer, machine learning, AI, exactly what we are playing with. And at the end of the day, you get a score. And this score is telling you whether you should go through chemotherapy or not. And if you don't go, I mean, if you don't need to go through biochemistry to chemotherapy, and you go, it is actually a disaster. And there are about 30% of the patients for which it is undecisive. If you don't provide the decision of a physician, the HER2 decision, but rather look at the slide itself and use our method in order to make a more refined analysis of the HER2 together with these genes, we suspect, I mean, we still need to process the data, but according to our preliminary results, we think that we can fine-tune this vague, this undecisive region in order to provide better results. We have other preliminary results, including cytology, which is not tissue, but rather liquids. And there is an interesting story that I would like to tell you. One of the head of departments, I will not say whom, we talked to, told us, look, if you'll find an error, a mistake in my department, I'm jumping out of the window. We started working together, and long and behold, his interns started closing the windows. In fact, they were not willing to tell him of some of our results. Now, it's not always their faults, because in some cases, when you do the slice wrongly or from the wrong part of the tumor, you don't see, there is no pathological evidence that it's there. But still, it was existing in the H&E, and training the system could actually see far and beyond what was accustomed right now. So this is an interesting story. These are companies in the field, which is really tough. And the next step for us would be really to try and push this technology out of the Technion, I mean, into a company. And there are directly related and unrelated companies that raise a lot of money, and it would be a challenge to two of them in Israel, the two at the bottom, to try to compete with them. We are really well funded in terms of Israeli grants, but again, taking it to the next step would be a real challenge. Now, I don't know how much time do I have, but if I have 5 minutes, if I do, you should tell me. 3 minutes, okay, so we can negotiate. You see how bad I am in doing business. I can tell you a little bit about the other stuff of my research. I mean, what do we do in computer vision that has also relations to what we do over here. So a while back, almost 15 years ago, we developed a 3D scanner in my lab that eventually we sold through a Technion joint venture to Intel. This is Muli Eden presenting the first sensor. It's called RealSense that Intel used, and they sold it. In fact, when you move to the industry, you learn that you need to optimize not only accuracy and time, but also the cost of this product. This is comparing to our competitors. And here you can see the output. So rather than just having an image, this device can give you the 3D structure. So at the top, this is the raw. At the bottom, this is after some processing. So this is what you get when you take pictures with this kind of sensors, which are really, really, really cheap and relatively accurate to their cost. And this is how it was used. I mean, probably it is not integrated well, but believe me, it was used, and you can actually see the outcome on your iPhones. So there was a Swiss company that was acquired by Apple, and using similar technology in order to move your images. But from a medical point of view, we gave our system to a physician, Israel Amirav. Now he's spending time in the border between Ukraine and I think Moldova. Helping refugees. And what he wanted to do is design a specialized inhalation mask for kids. So he scanned a lot of faces of kids. We clustered them using something which is called metric geometry. And at the end of the day, we manufactured an inhalation mask that was really tailored for babies. Now, Israel is a clever guy. He also punctured a hole for a pacifier, which has dual effect. It also calms down the baby as well as create better sealing. But it appears that when we sent the system to Congo, for example, kids were not willing to take pacifier. They never heard about the stuff, so in some cases you need a version without it. This is how it looks like compared to the old version of the inhalation masks. I can tell you that we also have a joint project with Alon Wolf and one of his students, Benjamin Greusser, collaborating with many hospitals here in New York in order to define sclerosis in adolescents. So we take 3D image of your back, and we know how to predict what is going on and what is wrong. And there is, again, a lot of really high-quality data that we are processing. This is yet another project in which we take a regular image and based on deep learning, this kind of black box that I was talking about at the beginning can produce the geometry that you see here on your right. And on the right, I mean, there is center and right. On the right, you see the image itself being texture mapped onto the geometry. This is something that we did about 4 years ago, and people are still trying to compete with us, and I could say that we are still winning. So the students were doing a really good job. This is, again, it's not me, it's the students. I'm not to be blamed, but I will communicate that. This is more or less it, so thank you very much for your attention.
Last night at the dinner, Walter Isaacson said he thinks the biggest risk to society has today is social media. And in 1950, before he passed away, by the way, George Bernard Shaw said, "Beware of false knowledge. It is more dangerous than ignorance." And I think social media is making more use of computer vision, machine learning, the ability to twist images, to easily demonstrate that they can present to somebody an image, a voice, a face that is simply not real. My question is, what are all of you people at the cutting edge of machine learning and artificial intelligence doing to make sure that the media itself is not going to continue to make it impossible for the average person to know the difference between real facts and total bullshit?
This is an excellent question, and I didn't talk to it before. Okay, so again, one of my students, Michael Bronstein, who is now a chair professor in Oxford University, has started up a company using metric geometry methods combined with deep learning. The company was called Fabula, in order to detect fake news. It appears that there are patterns that characterize fake news. So I wouldn't say that it is perfect and it is bulletproof, but you can use deep learning, non-traditional deep learning, in order to find these aberrations. Now, I would say that every news is fake, in a sense that each one writes his own history, so there is no limit of what is fake or what is. So we need to keep the boundaries right. I agree with you that we need to learn technology, and then we'll have analytic tools to decipher what is true and what is not. If it doesn't make sense, if it doesn't add up together, then the math would tell us that this is irrational. And indeed, I mean, Fabula was eventually purchased by Twitter, and he has a group over there, and they are doing fine. So, and again, I didn't invite this question. Thank you.
Thank you.
Thank you again.
Shai is the last one that will present.
Thank you all. It's really an honor to speak to this crowd, and thank you for the support that you've been providing to Technion for all these years. My purpose in my talk today is actually twofold. One, I'd like to tell you about the revolution that's happening in immunology and how it's impacting health. I think you've experienced all of this in the past 2 years, but there's more to come. And the second is to show you how in this particular field, how multidisciplinary work has been going on together, and how this basically reflects back to what Uri told us this morning about how the Technion is changing to fit the 21st century needs. So before I really start, but actually an example of what I just said, here's my team, my lab. Just in this little group of people, you have biologists, computer scientists, electrical engineers, physicians, MD, PhD students, and nurses. All of them are needed to be able to get any one result out of this lab. And together what we've been doing is really kind of trying to tackle two different questions. The first one I think is something that's relatively easy to relate to, especially in the past 2 years. And it's this question. Imagine you walked into a physician's office and you said, "Hey doc, how's my immune system doing?" Sounds like a simple question, right? So just to kind of give us a frame of reference, right? If I were to ask the physician, if you were to ask him, how's my heart doing? There's a lot of different assays that the physician can give you. There's a lot of different interventions that they can do, either lifestyle or surgical and so forth. But for the question of the immune system, it's not really trivial. In fact, it's to the point where if I wanted to just ask, do I have a good immune system, or I needed to kind of try to say, order the people in this room from people who have a wonderful, young, healthy immune system to one that may be a concern, and I'm going to put aside, say, people who have extreme illnesses where it's kind of trivial, it's not going to be an easy thing to do. I can certainly order people here by height, but ordering them by immune system health is not something that we know how to do at this point. And this is the system that's in charge, basically, of every disease, of handling every disease we have. And some diseases, it's the thing that goes wrong so that basically the disease breaks out. So it sounds kind of almost as important as knowing what your heart status is, but we don't have an answer. Right? And the reason the physician basically has no clue on this is because this is actually one of the most fascinating systems in the human body. It's a sensory system, and it basically continually evolves and learns from your life history as you live. I basically think about it as a sixth sense. Okay, basically you just think you have something, you have a system in you that continuously monitors your external and internal environments and adapts to them so that they can best protect you. And sometimes it goes wrong, which is a problem as well, right? So it's an extremely complicated system. It's a cellular system. It's a distributed system in your body, many, many, many different cells that together give this emergent phenomena that we call immune response, okay? So for many years, just trying to tackle this question of how's the immune system doing was just basically beyond comprehension. In fact, we can go back a decade or 15 years, totally science fiction. And the reason is because the human immune system is so complex, we didn't have the technologies to be able to get a sense of this. And if you go back to 2007 when I started my postdoc, I had this idea that, well, maybe it's time that we could actually study, do basic research on the immune system in an area that's actually translational, impactful. You can do human immune monitoring. Technologies seem to start being there. And in the past 10 years, or as I said in the title of my talk, this is a field that's been coming of age. And we've been developing the technologies that allow us to give the big picture, to get the big picture about the immune system. Every cell we can now quantify, we can start understanding how they come together to yield this response, and we can start understanding how each one of us varies on our immune system status, and how that basically then reflects on clinical events, whether it's disease severity, or responding to drugs, or vaccine responses or infections. So that's really been an amazing change. And the 2007, basically, technologies are not there to do it. Now, every layer of your immune system biology, we have at least one technology, and sometimes way more, that can pretty much not give you everything that exists there, but comprehensively measure it. And it can do so in humans, and it's still expensive, but the prices keep going down. Okay, and so we can start studying human immune variation and trying to tackle this problem. So we've been doing this in the lab in many different areas. Most prominently, we asked the question of how are we all different as we change with age, right? Because the biggest change in the immune system over time is just us experiencing life. And so if you look at children, where we see immune variation, but relatively small, as we age, as we live through life, the immune system variation between us keeps growing bigger. And in the end, we even know this, we know some people have a better or worse immune system. We have a sense of this, right? So understanding if we can quantify immune variation with age, that would be something that would allow us to define immune health. And then understand from this how this relates to the disease. So we've been tracking people longitudinally back from 2007, continuing today. It's a large project funded by the NIH, where we've been monitoring longitudinally people with the latest technologies and understanding how the immune system varies. And from this, we've been able to define something we call an immune age of a person. And now we're studying how this relates to severe infections such as sepsis, how this relates to inflammatory bowel diseases, can we predict drug responses in autoimmune diseases and in cancer and so forth. So this is the basic idea of what we've been doing in the lab. And just to give you one data figure from our longitudinal study of aging, this is from the immune perspective, this is the longest running, highest resolution study of the immune system. Here's one data slide. On the x-axis, you see older individuals, 16 up, okay? On the y-axis is the frequency in blood of a cell we call naive CD8 T cell. Okay, it's a cell, probably you never heard of it before COVID, maybe you heard of it after COVID, but basically this is a cell, it's one of the hallmarks of immune aging, it drops with age, okay? And on the black line you see is if you take the population average, how does it change? Well, it goes down with age. It's been reported in hundreds of papers. But the fact that we've been tracking people longitudinally for the first time with this study allowed us to understand how does this vary between each one of us. And the colored lines that you see here are each describing how the cell changes in any one person. Okay? And what you can see is actually we have very different rates of change for naive CD8 T cells, and sometimes people are actually going the reverse direction than what we would have expected. Okay? So this is one cell, and I said we have the technologies to measure a large number of cells, and in fact in the study we've measured over 120 different cell types, immune cell types, each of which has a different function, each of which plays a role in emergent response. And so we could quantify for every cell and over 150 people, what is the rate in which the cells are changing? And rather than thinking about it one cell at a time, think about the multidimensional behavior of the immune system in each one of our cells, and so in each one of our bodies. And so when we did so, we realized that what people think of as being very different immune systems or kind of different patterns in the immune system, in fact, I will say here that if we measured everybody's blood, we did COVID testing, but if we did blood testing here, this is like a fingerprint. I can identify a person based on their immune cell composition. Okay, and so high resolution, way more than you get in your HMO for now. This type of profiling of the immune system, rather than, despite the fact that we all look unique, now that we've tracked people longitudinally, we understand how we change. And it turns out we actually change in a very similar manner. And the reason we look different is because we do so at very different rates. It's like I'm driving on a road from point A to B, and we're all driving in different cars at different speeds. And I call you up and you say, what do you see out the window? And you describe a landscape. It doesn't look like mine. We must be on a different road. But in fact, if you wait long enough, you'll find out that I'm going to get to the same landscape, okay? So this is the basic idea. We have a pattern. We can quantify where you are on the road. And just to kind of show you the kind of a sense of this, it's basically a relationship, a positive correlation between your chronological age, how old you are, right, when you were born, and your immune system age. And it's a noisy positive correlation. So basically, we can look at this graph here, and you can see that for any given person, this is the actual data slide is too complex to show, so I'm showing a cartoon of this, but basically for every person, we can now take immune cell frequency, or a set of 49 genes, and place them on this trajectory that describes their immune age. And it turns out this not only describes functionally how your immune system behaves differently, but also clinically is relevant. So what we did is we took this signature that gives us an immune age score, and we went to the Framingham Heart Study. This is the largest cardiovascular study that's been run in the US since 1950. Every person there, about 10 years ago, got measured for a set of genes that we could take our score and assign what is their immune age for every person in Framingham. And what we were able to show is that this immune age information captures information that is not available in any clinical database that describes the Framingham Heart, that describe the patients in the Framingham Heart Study, and it provides additional clinical benefit in predicting survival of patients. Okay, so we can provide information that just isn't captured in today's electronic medical records just by quantifying the immune system. And since then, we've shown this affects how you predict drugs, trauma, patients in trauma and in sepsis, cognitive decline in Alzheimer's, and in other places. Quantifying your immune system state gives you value. So this is the kind of the first direction of this, but it's one, if as you've heard me talk, you probably noticed one other thing. I'm talking about a lot of data, right? So the other side of my lab works on what I call the data insight gap. Basically, since the 1990s, and this is true not just in healthcare and biomedicine, the we've moved into the big data era. Data is exploding. It's an exponential stage. And the problem is that insight and kind of what you get out of the data isn't following the same slope, right? So basically, every day we get more data, and the insight, the gap between how much data we have and how much percent of information we get out is actually decreasing. So this is the data insight gap. So the other side of my lab works on this problem. And you can think of it, how do you solve this, right? So put a lot of computer science in, and to give you just divide algorithms to basically get more bang for the buck. And just to give you a sense of something I think we've all experienced in the past 20 years, this is what happened in the phone revolution, right? So 25 years ago, analog phones, no data whatsoever. We moved to the Nokias and so forth. We had big data. That GPS, not very useful for us as phone consumers, right? Maybe good a bit from the technical side of the back end. You take that same GPS data on a phone that models the real world, say a map, and suddenly this is a device that we don't leave our home without, right? So the same idea is what we've been doing in biomedicine now. We've moved from, in the same 25 years period, we moved from no data to huge amounts of machine-readable data. You also heard this from Juan. And what we need to build, and what we've been building, is a suite of algorithms that allow us to think about all the data that we have in a disease in one disease model, okay? And so this idea is extremely useful, and we've actually spun it out into a company, but I'll show you the biggest useful utility we found out of it. And basically this is, when we do drug development is extremely risky, extremely difficult, and so forth. Why do we start at the beginning as if we had no knowledge of what the disease looks like, of what all the information that's previously come before? Instead, we can take all the world's information about this disease, all the literature, all the gene expression data, the proteins, and so forth, wrap it up together in a single model, and then push that all the way to the beginning of the trial to make a choice of what should we be targeting, which disease should we go for, what's the right experiments to do, move drug development from 8% success rate in phase 1 to on the order of 24%. Right now. This is the estimate of what happens when you start using big data early stages of drug development. So this type of ideas are actually totally changing cause us to rethink drug development. And in the same type of ideas is what Uri told you this morning. We need to be rethinking about academia. And so what we've been doing over the past summer, Uri pushed us into a process where we started thinking multidisciplinary. How do we take the Technion to the next challenge? How do we address undergraduate education, graduate education, and faculty recruitment and research in a new multidisciplinary environment where it's clear to us that we need to be pulling faculty from the different disciplines into doing something that's tied to the large challenges, human health, sustainability, the two most relevant ones for the 6 life science faculties at the Technion. We formed committees that basically looked for almost 4.5 months at reevaluated how we're doing undergraduate programs, kind of looking at merging programs, what new programs do we need, how do we tie this to graduate education, how do we recruit multidisciplinary faculties that don't fit, kind of new candidates that don't fit any one faculty but that would feel like they have a home, and most of all, how do we take our own individual skills and excellence and so forth and put them together to work on big challenges. And what are the big challenges that the Technion would be best suited to work on in this domain? So we need to evaluate our strengths, our weaknesses, and so forth. To do so, we came up with a bunch of different solutions. I'll show you kind of a bunch of different focus areas for research. We divide them into centers that fall under our 2 engines, the Human Health Initiative that we mentioned and sustainability. So those 2 initiatives, below them we see centers being formed. Some of them we kind of knew ahead of time, such as cardiovascular medicine. The Technion has almost 30 faculty that work in this area, but others are tech-enabled science, taking the people who are asking basic questions in biology and putting them together with the people who are developing the latest technologies to answer those questions. Neuroscience and neuroengineering, food tech, aging is being another topic in which there's many researchers that are working from different angles of a huge problem for society. We can now put them together in a single kind of institute or center and start drawing the power together. And also themes, small centers of excellence of 3, 6 faculty members that we know are focused on a particular area. They make a huge impact, and we want to put them in the same physical space or in the same resources, such as ubiquitin-related diseases, as is the Nobel Prize. And so this is some of what we've been doing in the centers. On the bottom right here, you see modeling and data for life sciences. It's basically merging this computer science into kind of the biomedical domain. And for this, we've actually already initiated some of the efforts. And I'm just going to show you a small movie from a video from a datathon we held last week.
Hackathon is something really new in between Rambam and Technion. It's our first event like that. I hope that there will be many more.
The way to foster collaborations is through events like the event that we have here today. We have Rambam IT. We have Rambam clinicians on the ground. We have students from, I think, 5 or 6 faculties and researchers from the Technion. The clinicians bring their side, which is formulating the problem and the domain knowledge. The students and the researchers from the Technion bring their side, which is how to build algorithms and analyze big data, and together we'll find solutions.
This 3 days event, including a 2 days datathon, is inaugural event of the new center. CAIH will be the new Israeli academic center for medical AI, dedicated to basic medical and clinical research to improve patient care.
There's several outcomes that I want to see. First, more and more collaborations. The second is to have ongoing development of models and their deployment at Rambam to test them. And ideally, in a year from now, we'll have tools on the ground affecting patient lives and outcomes.
This is just from last week, more to come. And I hope I showed you in my talks, both in the lab and what's happening in the Technion, that basically where we're moving to is a lot more working together within the Technion and with industry and so forth. And thank you.
The last question. You showed very consistently the progression of the immune system and pointed out you can tell all sorts of things about the inevitability of people getting to these places. Optimists that I know, and more of them in the clinical research community, are saying aging itself is a disease state and not inevitable. And many of them point to looking at the immune system as sort of the metric by which we age. Do you believe, with all the other things we can manipulate these days, genetically, CRISPR and all that, do you believe that we are in anywhere near the reasonable future going to be able to use the immune system as a mechanism to reverse aging?
It's a wonderful question. Obviously, we think about it a lot. I'll say, so here's the good and bad news. Good news, we already have early evidence that we can basically push back immune aging. So we can, using a drug, we can take people who are responding to the drug, not everybody responds, we can kind of reverse it. But what we don't know yet, and we suspect would be difficult, is to keep it young. Because when you think about aging, it's a systemic problem. And so if I've made my immune system younger, but it still lives in an environment that isn't, right? I need to re-educate the environment, and how I do so isn't trivial. So I think the bad news is going to take a bit long, and what we are doing right now is taking on these same people that we've just used to just measure their immune system. We are now measuring aging on almost every kind of bodily organ to try and see how this fits together, where the immune system is making where that interaction can be manipulated so that not only the immune system becomes younger, but also it changes the organ functionality as well.
Okay, you guys can all have basketball. This is March Madness right here. I can't thank Dean enough for being the moderator, for your amazing words. Tamar and Ron and Shai, I see a lot of collaboration, and it's so wonderful. And this is why I support the Technion, and that's why all of you do too, and spread the word.